Hello everyone, I am Liu Hongbin from Shandong University. Today, I will share with you that how to prepare the mouse models for gene editing in reproductive medicine and also its related application. Hopefully, it will be valuable for your future research work. In vitro models often cannot reflect the real situation as in vivo. For example, the ovarian granulosa cell culture, which are luteinized granulosa cells, and cannot accurately evaluate the effect of the target gene on follicular development and ovulation. Similar, Sertoli cells and spermatocytes cultured in vitro are truly unable to reflect the regulatory function in spermatogenesis as in vivo models. In addition, in vitro experiments tend to amplify or miss the actual function of genes, and their physiological significance cannot be measured accurately. Animal models are crucial for the human disease study in reproductive medicine. Using animal models such as zebrafish, mouse, and monkey, the function of specific genes and the pathogenesis of disease can be clarified, with the potential therapeutic targets can be discovered. This can provide a theoretical basis for the diagnosis and treatment of infertility. Scientists have been exploring more efficient gene editing techniques to alter the expression of specific genes in animals. For example, knocking out or knocking down target genes, overexpressing a target gene and inserting one gene. Currently, we focus on the mouse models, which have the following advantages. As genetically engineering mice, the mouse and human genome have the higher homology, easy genetic operation. The whole genome has been sequenced, and the mouse genetic background is clear. Small size and easy to feed. Short reproductive cycle. We generally using the following three strategies to study the in vivo function of genes: conventional knockout mice, conditional knockout mice, and knock-in mice. If a gene specifically expressed in germ cells, conventional knockout mice can be constructed, such as GDF9, a specifically expressed gene in oocytes. Knockout mice resultant female mice are infertile. Another example is the testis-specific gene of TAX11. Knockout of TAX11 gene causes male mouse to sterile. However, for genes that are expressed in all tissues, or the conventional knockout mice is lethal, it's necessary to adopt a conditional gene knockout strategy to study their function in the reproductive system. Clinically, by studying the genetic cause in families associated with infertility, discovering a pathogenesis mutation, comparing the homologous genes of humans and mice, finding the position of homologous mutations, and constructing gene knock-in mice carrying human homologous mutation sites, and to conduct functional studies of the disease. This kind of mice is commonly constructed by the CRISPR-Cas9 gene editing technology, consisting of three parts. The first part is the Cas9 enzyme, which has two domains of nuclein activity and is used to cut DNA double strands. The second part is the sequence-specific single-guide RNA, which determines the binding gene target and mediates precise cleavage of the Cas9 enzyme. The third part is the PM sequence, which mediate the binding of guide RNA to the target site, which is necessary for the activation of Cas9 enzymatic activity. Its working model of action is as follows. Under the guide of the guide RNA, the Cas9 enzyme cuts at the target sites to the gene of interest to generate DNA double-stranded breaks, which are repaired by the joining of non-homolog end. Where DNA breaks, insertion or deletion of fragments of different lengths can occur, resulting in the loss of the target gene function. The technology has obvious advantages: low cost, high efficiency, wide coverage, and simple system construction. For genes specifically expressed in the testes or ovary, conventional knockout mice can be constructed by Cas9 technology. Taking the testis-specific expression of the DCW-PW1 gene as an example, two single-guide RNA are designed at both exon number 3 and exon number 7 to achieve large fragment knockout. This large fragment gene knockout method can effectively delete multiple transcripts, ensure the loss of gene to study the mechanism. By constructing DCW-PW1 gene knockout mice, it was confirmed that the epigenetic regulation factor of DCW-PW1 gene play an important role in mouse meiosis. These are differences in meiosis between male and female mice. In male, DCW-PW1 knockout mice have shown the features of azoospermia. However, female mice have shown primordial ovary insufficiency. We also found that the SRE gene, the reinforcing element of the synaptonemal complex, acts as a button to maintain the integrity of the synaptonemal complex. In SRE knockout mice, male mice indicated azoospermia, while the female mice have shown primordial ovary insufficiency. This section describes studies in conventional knockout mice, and in the next section, we will describe the studies in conditional knockout mice. Thanks to all of you for your attention.